Really happy to have Ashwin here with us today. He's an early career researcher and has been passionate about bats, and you worked in the middle on dragonflies? No. Spiders, and now he's been working for the last 5 years on birds, but he's really driven to understand questions in disease ecology. So it's very nice to see, because as you can see already, it's a very complicated topic which is so interdisciplinary. And sometimes early career researchers might feel that, you've got to just get ahead and succeed and all. So maybe you shouldn't take on something very complicated, but he's definitely done that, and it'll be great to hear from him. Thanks, Ashwin.
Thanks, Uma. So, hi everyone, I'm Ashwin. I've talked to quite a few of you in last 2 days. And I've been working on avian malaria in birds, other hemosporidians also. And today I'm not going to talk much about my research per se, but trying to have a more general lecture on how this system of malaria and other related parasites in these birds work. Although it's investigating pathogens in birds, I'm not really well versed with the other pathogens in birds. So just one slide on other pathogens, and all the slides are on avian malaria. So sorry for that. And yeah, since we are before lunch, I hope you'll get hungrier. I'll make you hungrier with more and more complex systems. So a very general this thing that for emerging infectious diseases, we have, in most of the infectious diseases, it's like a host and parasite continuum between wildlife, domestic animals, and humans. And what happens most of the time is a very nice review and very nice representation that whenever there is a close proximity of humans with wildlife, or humans and domesticated livestock with wildlife, that's where the chances increase a lot. That's when you have more chances of getting, there is a spillover. It's a very interesting concept, the spillover, that why and how it happens is when you have a reservoir host where the parasite is residing in that host for a long time, and probably it's in an evolutionary equilibrium with that. It's in a co-evolutionary cycle, and sometime it somehow manages to get into another host, and it finds that host very appealing, very livable, and that's where it starts growing very fast, because perhaps that host does not have mechanism enough on an evolutionary arms race to deal with that parasite. And that's when you have these peaks. All these outbreaks that have been happening are a lot of times something like COVID also. We have outbreaks where there is a spillover, and that's when the peak happens. And there are again a lot of there are disturbance-related factors that are caused by human activities that make it more and more plausible to have these spillovers. But what is important is if you have control operations, if you have an early detection system, if you are able to detect how many parasites are present in the wilderness, in the diversity of hosts that we have, and we know what are their associations, then it's easier to have these control operations whenever there is an outbreak before realizing that it's out of our hands. So that's what is very important. So host switching is on an evolutionary basis, and human activities on an environmental basis. There is an amalgamation of those two things. Host switching has been happening on evolutionary basis also, but human activities cause it on an ecological scale, and there is no time for co-evolution, and there's a breakdown that can happen. So the structure of this talk will be, I'll go through first quickly. Yes. Sorry? What is? Sympatric. Sympatric is there something sympatric here? So sympatric means when two species are staying in the same place. Like allopatric, when there is a barrier in between and two populations or two species are isolated. So they're sympatric species. If there is a bulbul and a sparrow staying in the same forest, they're sympatric species, or any two species. But if there is a Malabar whistling thrush and blue whistling thrush in Himalayas, are allopatric species, they are not really staying anywhere. There is no range overlap.
Here.
Hi.
Hi. Can we predict spillover using some model?
Yeah. I think the second time this question is happening. Yeah. I think if we know enough information of what are the parasites in the closest host possible, what are the possible interactions, and what is the genetic and molecular level compatibility of hosts. If we have enough information on that, we can have models, but we yesterday only saw essentially all models are wrong. But some of those can be useful that can help us predict. that can help us at least stay away from some of those scenarios to not even go for a possibility of spillover, right? So I'll walk through birds and parasites. I mean, birds as host, why birds, and why are we studying birds so much, and parasites in bird diseases. Then I'll immediately get into avian malaria. Then we'll go through how avian malaria system works, what are these parasites, what they do exactly in the bird's body. And then there are outbreaks, and there is something called abortive development, which is very interesting phenomenon. And then there is a very interesting case where the avian malaria came into light of wildlife conservation is because of an outbreak in Hawaii that happened almost a century ago. And then we look at factors affecting the parasite prevalence. What are these human-induced factors? What are these ecological factors that are causing those parasites to, have certain dynamics with their hosts? And a quick peek into the study that I have been doing for the last 5 years. So the birds and parasites are the first part. So why birds? Why are we so attending to birds? I mean, they are national emblems like dodo. There is a cultural, national, spiritual, and individual interests for all those. For example, these are called duck stamps. In US, these duck stamps are compulsory purchase for anyone who is going for duck hunting. If they want to go to any protected area for duck hunting, even if they have duck hunting permit, they have to buy this $1 duck stamp in order to enter the PA. And that has produced millions of dollars, which are used for wildlife conservation. I mean, these kind of also symbols that birds, even for hunting, they are very important, and people are interested and invested in birds. Then something like cultural aspects, we know that a lot of different cultures have birds as different symbols, different ways of celebrating birds or using their parts. And then of course food, tandoori chicken everyone likes. What else? When it comes to disease ecology, they are one of the most diverse group of vertebrate taxa. And I think one of the first things that we looked at was the parasite diversity is correlated with the host diversity, right? You have more diverse hosts, you have more diverse parasites. So when you have a most diverse group of vertebrate taxa, you can expect a lot more parasites within bird systems. That makes it even more interesting to explore these systems and see what is lying there. They're globally distributed and highly mobile. So birds are going everywhere. They're migrating, they are shifting their ranges like Umesh showed yesterday. They're so mobile. So the chances of these interactions with parasites and humans or between birds are also more likely, fairly predictable perhaps. And then they are one of the taxa that humans are living in very close proximity with, way more than mammals in terms of wild birds. We are not living in very close proximity with a lot of wild mammals, but compared to that, we are living in very close proximity with a lot of wild birds, including the feral pigeons have become feral now, but are essentially a wild species. And nobody, I mean, who knows how many parasites they are harboring. They are just literally nesting and pooping on our windows, in our balconies. And most important thing is we have a fairly good idea of the ecology of these birds. Since there is so much interest in birding, bird research and everything, we have a good idea of how the host is. Because if you want to look at the parasites, we need to really know how the hosts are behaving, how the hosts are acting and responding to their environment. Because the parasites are not free living, they are dependent on hosts, while these hosts are free living. And we have a fairly good idea of how the birds that hosts are in terms of their ecology. There are quite a few pathogens actually. There are very nice books that you can go through, and just at the start of this section, there is a book that I showed. There is a very nice good description of different parasites from bacteria, viruses to protozoans and even fungi in birds. But what comes to regular limelight are protozoans, which a lot of them cause avian malaria or malaria-like disease like Plasmodium, Hemoproteus, Leukocytosis, Toxoplasma. Toxoplasma is also found in birds. And then Trichomonas, Isospora. Then there are bacteria like Salmonella. Then there is Chlamydophila psittaci, which causes a disease also in humans called the parrot fever, which is not now it's called as I think ornitho fever, because now they realize that it's not just parrots, many other birds are also carrying this particular pathogen and that can jump into humans. Then there is Mycoplasma, TB, cholera. The strains of TB and cholera are also found in birds. And there are viruses like pox virus that Umesh yesterday talked about. There are these legions on these birds. So mosquitoes are actually mechanical carriers. So there is no life cycle inside mosquitoes, but it's a contact that they are spreading that virus and into other birds, and they are able to multiply, but this is more mechanical contact even with very close proximity. And if you are handling birds and if there is same handle, let's say a pox infected bird is handled and I don't wash my hands and I handle another bird, there is a good possibility that perhaps the pox virus can enter another bird. You have influenza viruses. We all know how much we are already tensed about this virus getting jumped into humans. Then there are herpes and papilloma. So these are the clear signs of papilloma viruses. If you see sparrows or sometimes larks near garbage dumps, you'll see a lot of times their feet will have very scaly skin. It's very dry and scaly skin. We have also caught some of these birds, some blackbirds that had very scaly skins. It's not a very smooth leg, but it's very flaky. And it starts bleeding immediately. I mean, it's so dry and so scaly that it starts bleeding immediately. And then we realize that these were all papilloma virus infections. So these ones are the actual lesions that Umesh was talking about. The eye almost gets covered. Eventually, birds may not even able to see how it is, its surroundings properly. But now getting into the host-parasite relationships. I mean, we all know that parasitism is a negative relationship where host gets negatively affected, parasite gets positively affected. But it's a very simplified definition. It's actually not that simple, but it's a very complex co-evolutionary process between the parasite and the host. And there is an evolutionary arms race, right? The Red Queen hypothesis that a lot of times we come across, that parasite is evolving, host is evolving to cope up with it. So it's an arms race that is constantly happening. And we'll come back to that, what that arms race could have happened. If you stop that race, what can happen? If you just take out one of the competitors from the race, what can happen? Parasites also impose selective pressures. I won't take a lot of time on this, but there is a very interesting study where they have shown that in one of the nematode worms, a bacteria can impose a selective pressure for outcrossing. Whereas when they had a control situation, the outcrossing went down. But the outcrossing did not really help those systems. But whenever a very co-evolving bacterium was introduced in those nematode systems, that created a very high selective pressure for outcrossing. Because if whatever that were inbred, selfed worms, they did not survive. So even only those that could outcross survived because they could actually co-evolve with the parasite by exchanging genes from multiple individuals. How does this host switching happen? Host switching happens whenever this kind of parasite systems have, and we have host phylogeny. We have hosts which are co-evolving with them. One can be association by descent when there are, through a strictly co-evolution as per the high specificity of the host. So there is a host and there is a parasite, and they have been evolving for millions of years. So there's a very tight co-association between them, and it is very less likely that those parasites will switch host. And then there is a horizontal transmission where there can be host switching. It can be environmentally, it can be human-induced, it can be also evolutionary. And something that a very easy way to look at it is when you have two phylogenies, you have a host phylogeny, you have parasite phylogeny. If you try to just kind of overlay one above the other, if they're following the exact same pattern, then the associations were very tight. But if that is not the case, as you see here, this is the trajectory, but this is where the parasite has diverged at a different time point. And the association is different. So now there is no direct, a congruence between the parasite and the host phylogeny in their evolutionary trajectories, which these phylogenies are kind of showing. And it's a nice statement I found that microevolutionary assessment of extent of and departure from co-speciation are necessary to define limits of associations and determinants of distribution and structure. It's just very nice, fancy statement I found. I thought worth thinking about. And then avian malaria. Yeah.
What's the difference between host switching and non-congruence?
So non-congruence is, actually these two are not very different things. Non-congruence is when you're trying to look at the system, the way parasite has evolved is not completely dependent on host. There might be some other factors that could have happened. It's a very hypothetical scenario. There's nothing, I'm not presenting a hard example from any wild systems, but this is a hypothetical scenario. And host switching is when, evolutionary trajectory took host to one side, but the parasite managed to somehow, diverge and managed to infect another which was not in the same evolutionary trajectory. A distantly related bird or distantly related host is when that host switching happens. It can be chance events. Most of the times it would be chance events when it will happen, right? Low adaptation?
When it switches to a new
It's less adaptable compared to the congeners.
Well, if it manages to survive in the new host, then we can't say it's less adaptable because it actually manages to adapt to a new host also when it was already infecting another host or its ancestor.
Okay.
So probably it manages to have enough adaptations to be able to switch and survive. It wasn't a dead end for that parasite, right?
Yeah.
Does similar relationship is there in vector and parasite, like, or it
so
is
vectors-
in host parasite only?
They should be, because vectors are still intermediate hosts, and even in case of avian malaria, not every mosquito is transmitting every parasite lineage. There are some mosquitoes only they are able to. There is a competence between host, the vector and the parasite. So, yeah.
I was just wondering if, just to clarify your point, does it make sense to just look at the trees and not just the arrows? Because the relationship between the parasites are different, on the left side and the right side. So that's the big difference.
I want to discuss some of these things tomorrow as well.
Thanks, Janvi. So now I'll come to avian malaria and other hemosporidia. So generally avian malaria, when we say malaria, it is regarded, it is with reference to Plasmodium infection. And there are other parasites which are very similar to Plasmodium, but malaria is whenever any in reading also, if you come across malaria, probably those that is being referred to only Plasmodium. So it's always called avian malaria and malaria-like parasites or malaria-like diseases. So what is malaria or these hemosporidians? They are protozoan, they are blood parasites. And for transmission, they require an intermediate vector host, and that's an obligate vector. It's must. Otherwise, two host will not be able to. There won't be any transmission between hosts if the vector is completely absent. And these vectors are almost always insect vectors. But different vectors, different taxon between insects, the dipterans, for different parasites do exist. They're known to infect reptiles, birds, and humans, all of us. And when they are in pathogenic form and when they cause that pathogenesis in the host is when the term malaria comes into, because malaria is a disease, is a symptom, which we are calling it that symptom, and the host is suffering enough, then when the malaria as a term comes in. Until then, it's still a host parasite association. So pathogen and parasite, that's where the links or non-links are. And there is a good value for human health because when malaria was being studied, when people were dying with malaria and we had no clue as humans that what is happening, one of the first systems where it was studied and it was shown that mosquitoes are ones that are carrying these microbes from one host to the other, that happened through birds. So it was birds that were the model systems to study malaria until very recently, until almost 1960s or 70s, when they found malaria parasites in the other primates and rats, is when they shifted from birds. Until then, birds were used as the model systems to study and experiment with malaria parasites. Very interestingly, that discovery happened in Kolkata in India, by Ross. What about the mode of transmission? How many of you have heard of this, mode of transmission of disease? There is this density dependent, with respect to host densities. So density dependent and frequency dependent. Can anyone just take a guess that can malaria be density dependent in terms of its transmission? So I'll quickly tell you what density dependence is. Simply like if there are more number of hosts, there is more likely of transmission happening. You increase the density of hosts, that means more hosts packed in a smaller area or per square kilometer, per square hectare, then there is more chance of infection spreading, transmission happening. So do you think malaria is density dependent? Can anyone just take a guess or informed guess? Yeah. No? Yeah. One no?
I think it's something to do with the life cycle of the parasite inside the vector than dependent on the density of the host.
Right. Same. Anybody else has a different opinion? They think it should be density dependent? Huh. Yeah. Yes.
Frequency dependent.
But right now I'm talking about humans as host or birds as host.
So we talked about frequency dependent, right?
Right.
So what is that frequency dependent?
So that's what I'm coming at. What is frequency dependent? So
So
there are two types of responses in disease transmission. One is density dependent, where the contact rate between susceptible and infected individual is directly correlated with the density of the host. You remember the COVID parties that were happening? Why those COVID parties were worse for even spreading COVID further? They're packing individuals in one small hall, they were partying, and it was spreading it further ahead. One infected individual could spread it to more number of people if more people are packed. And that's why we had social distancing. You can easily have malaria parties and stay away if you have mosquito nets around you. That's a very simple way of saying how frequency. Yeah. In somewhere, I'm not sure. But I mean that way. So even the mosquitoes' ability to spread it to further other people or other hosts is also very limited. I mean, I sneeze once, and if I have COVID, I sneeze once, and it can literally spread to 10 of you. But if you're all sitting together, it's a small room, it can easily spread to 20 of you. That's enough. But even if there are a lot of mosquitoes and I have malaria and it bites me, and then it won't immediately bite you and transmit malaria. The
Yes.
malaria parasite has to go through.
Yeah, and it is like pathogenesis of the disease, which is deciding transmission, whether in blood, whether it is transmissible or not.
Exactly. So the mode of transmission also has a big impact. So this is not density dependent. So even within mosquitoes, the parasite has to go through some life cycle. There is a sexual reproduction. It's not like the mosquito took up blood and next minute it's ready to transmit it. It's going to get few days. Sometimes it's up to a week or 16 days it can take for it is to be ready. And meanwhile, the party might be over, right? Will be over. So in that sense, it will be very frequency dependent in this case, that how frequently the vector is managed to, to spread it between humans. The more number of vectors and overall the bite rates are very high and those are not being taken into account, then the frequency of carrying that, even if there are less density, the vector can manage to find different humans even at distant places and can manage to transmit, but with certain lag. So that's called a frequency dependent response. Yeah.
So when you're modeling this, like SIR, you consider density dependent as S into I, like how many infected are coming into contact with susceptible in case of density dependent, right? So your DIDT term will have a S into I.
Right.
But how do you model frequency dependence? Like how?
I will share a link. There is a little complex math. There are some parameters that need to be they need to be derived. It's not as easy as density dependent, where you have a clear equation to model it. So for frequency dependent, when you're saying for frequency dependent, it's a slightly complex model, but there are models available to actually model the frequency dependent response of a disease.
So do you consider the vectors into the
Yes, you have to consider vectors into it.
Another thing, so I can classify this into vector-borne infectious diseases and non-vector-borne infectious diseases. Like COVID-19 is non-vector-borne, right?
So vector-borne infectious diseases would be frequency dependent, less density dependent.
Right. Okay.
Depends on again how the parasite is acting within vector. If there is any parasite that is readily, without going through any cycle within vector, there is no lag period. It bites me, it bites you immediately, and there is a transmission. Then the things differ.
So the time lag.
Right. There is also hunger, right? I mean, how much one vector will feed upon? It will get full with 1 or 2 blood meals. It won't go on biting everyone in this room, right? So that's also very important. Is everyone clear? I mean, okay with it? Yeah.
Frequency dependent, it would be a function of the vectors, the count of the vectors, and then we would
You're right. Yes. Please don't ask me about the math, but you're right. It will be a function of the vector, right? Okay. So these are the avian hemosporidia, the avian blood parasites that we generally deal with. There is Plasmodium, which is transmitted by mosquitoes. Then there is Hemoproteus and Parahemoproteus. These are two subgenera under Hemoproteus supergenus. They are transmitted by biting midges of the family Ceratophoganidae, which is different, still a dipteran vector. And then there is Leucocytozoon, which is a third blood parasite, which is transmitted by black flies. And there is also something called as Folicia, which is a shared parasite between reptilian malaria and the genus is shared between reptilian malaria and bird malaria. Like Plasmodium is shared between humans and birds, but it's not shared in the sense shared. It's just that it's a common genus. But the bird plasmodium cannot infect humans. So far, there are no cases at all. They are evolutionarily very distinct, but they're still in the same genus. The way their life cycle goes is very similar. So Faulicia is actually, we have no clue whether the vectors are really mosquitoes because there are not many instances. It is found from very few studies from North America and like literally handful of cases where Faulicia was actually detected. There needs to be a lot of more research whether how it is actually doing, and are mosquitoes are actually the vectors or something else? We literally have no clue how they are spreading in birds. Very few. Huh?
Is that mortality in case of birds, malaria, and human malaria, is that same or?
Yeah. So he asked if the mortality.
In case of birds, malaria, and human malaria. So is that the same or different?
Well, malaria, birds in mortality, it differs because a lot of bird species are co-evolved with the parasite and they are able to tolerate the infections. They don't die out. The mortality is very less unless there is a host switching that I'm coming to.
Because in case of humans, we have a treatment.
Right.
Whereas in birds, it's very less chance for you to get that infected before it's going to die.
True.
So it's a frequency of getting positive bird for malaria, it is very rare event. If the mortality is very high, it's a very rare event for you to get the positive, malarial positive bird. But if the mortality is very high, like that of human beings, so to study in that condition, it is very less.
So you're right, and we are getting a lot of birds which are infected. Because if the mortality would have been very high, we have actually got birds which are not infected. Because the birds that are infected are dying. We are not even sampling them. We are not able to find them. They're dying in the forest directly. But that is not the case. We are getting a lot of birds which are infected, which kind of an sign that there is a good amount of co-evolution happening. There is no mortality at least happening at a large scale, except for few examples, which I'll come to. And the mosquitoes are Culex mosquitoes, the Culicidae mosquitoes, and very few instances where some of the Anopheles were experimentally shown to be, capable of transmitting, but no direct evidence in wild. Plasmodium. Yeah, please. Sure.
So, if I understood your question was that it's very difficult to sample birds which have malaria. That's what you're saying. Yeah. Okay. So I think you are looking. So there are different phases of malaria. I think he will cover probably. So something is called acute infection, right? When you have a really full-blown infection going on, and there's something called chronic infection. Acute infection is a phase for a host when you are actually going through the symptoms of the disease. So those birds, if we are trying to catch, we will never be, because they'll be lethargic if they are experiencing any of those, pathological conditions. But these are the birds which we are catching in the field. They have had infection, they have recovered from it, but because the bird life is so small, they never clear the infection. It's going to be there. So these are like the chronic infections. So they are not carriers. Some of them may be symptomatic, but they have had infection. I mean, it does affect their life history traits, which I'll cover in the afternoon and all of that stuff. So these are not, these birds have the disease basically. So these are just a chronic infection because the life cycle of the bird is 4 or 5 years, or maybe some of them live 20 years or whatever. But once you get infected, it's very difficult to clear the infection. Does that answer the question? I think. Yeah.
Differentiate the bird which is carrying the parasite and also the bird which is not carrying.
If it is not carrying the parasite, you will not find the parasite. Okay.
No, can you differentiate? Just by looking at
this is why the methodology that is followed to screen the birds, you use combination of both microscopy as well as the PCRs basically. So, but if it's a blood-borne parasite, malaria has different stages. It goes through the organs. Plasmodium, if it is in the blood in your peripheral veins, which means the parasite is expecting to be picked up by the vectors. When it is in a very dormant phase, it goes into your liver or other organs. Basically, you will not find it in the blood. This is why the molecular work is important to help and to understand, even though parasite intensity might be very low. Yeah.
But there are no field symptoms. I can't look at a bird and tell if it has malaria or not. I have to sample blood or any other tissues. That's a dead. Yeah. So the parasite life cycle, like what Farah just said, it's fairly complex actually in this sense. And how the Plasmodium, I will just go through Plasmodium human proteus.
Hi. So can you move to the previous slide?
Sure.
Please. So you were saying the vector for avian malaria is Culicidae group, no?
Yes.
Culex.
Culex.
But in case of humans, it's Anopheline.
It's Anopheles.
Do you think there might be some difference in the adaptation or whatever? Like, I'm just curious.
Do you know there is an adaptation clearly? I mean,
there is actually, I mean, I was hoping to show that so these are like the vector traits which have kind of evolved in the time, basically. So there is so many different kinds of malaria, actually. There is rodents have their own malaria, bats carry malaria, humans have. But the whole diversification is actually driven by the vector species which are transmitting them. So Anopheles mosquitoes cannot transmit bird malaria, vice versa for the Culex as well. So, but the holy grail actually for that, which a lot of people are trying to work, lies in their salivary glands. Why are they not able to filter some of those parasites which others mosquitoes? And I think it's also maybe kind of a way of segregating their niche or how competition between the parasites, how things work. So yeah, it's the holy grail which everybody is trying to understand.
So evolutionarily very different, diverged long ago. So that with that, the adaptation could have happened with different mosquitoes, and now there is not compatibility. So distinct now. So what happens in Plasmodium is you have sporozoites which are injected into a bird's body through mosquito. Then they go under something called as exoerythrocytic cycle. It does not immediately go and attacks the RBCs, very similar to humans. But in humans, most of the, I think all of the cases, the parasite goes to liver specifically and stays there. And then it goes under those mirogony and then gets into the bloodstream and infects RBCs. With birds, it's not necessarily only liver, but many other organs, including bone marrow, spleen, brain, skeletal muscles, heart muscles. It can go anywhere. And it can go under its exoerythrocytic mirogonies. And there are two types of them. There is a pre-erythrocytic exoerythrocytic mirogony, where there are two types of cells called cryptozoites and metacryptozoites. I mean, it's just technical terms. No need to, it's a simple textbook reading that you will get it. All these cells are, or these stages are, their sizes are different and the type of organs that they infect. Reticular lymphoid macrophage cells versus the metacryptozoites can go to reticular endothelial cells also, including the brain capillaries. And then those metacryptozoites are able to differentiate in three different ways. One is they can give rise to more metacryptozoites. They can either become phanerozoites, which is another phase which stays mostly in the bloodstream, but in the capillary endothelial cells. And third is the erythrocytic merozoites, where they are actually able to infect the RBCs. And then these erythrocytic merozoites, when they rupture through the RBCs, they can either some of them will become gametes, some of them can again induce more phanerozoites. So this is the phase, the post-erythrocytic mirogony, that exoerythrocytic cycle is when there is an acute phase picks up. That's when there is a lot of parasites in the blood. There are also the RBCs rupturing. And they're giving rise to more exoerythrocytic phases also, and that's where the bird or the host suffers most. The post-erythrocytic mirogony is absent in mammalian malaria. So once the gametes are out, and this is one big difference between human malaria and bird plasmodium, human plasmodium, bird plasmodium, that some of these stages are absent in humans. And then the phanerozoites and endothelial cells of brain vessels take up the shape of vessels. They can enlarge and they can completely block the vessel and the blood flow to the brain, and that can actually kill the host, which is very similar to what happens in falciparum, right? The falciparum malaria in humans, the cerebral malaria, which is here. I mean, if you see this picture, so this is where the bird plasmodium is, and the way it happens, the functional aspect of it is slightly different, where the phanerozoites are actually they're enlarging, they're taking the shape of the vessel and they're blocking it. While in terms of the falciparum malaria, which is in humans, the infected RBCs are clumping together and adhering to the endothelial wall of the blood vessel, and that's what is blocking it. Is the RBC that are blocking it? Here the parasite itself is blocking it directly. So the symptom is very similar. Both of them die due to brain damage and the cerebral malaria, but the function is quite different. What happens in here, hemoproteus, is that there are not these cryptozoites and metacryptozoites, but there are these merons and megalomerons. So these porozoites get into these phases, and then there is no multiplication in blood cells. So the hemoproteus gets to other organs, non-blood organs. There is no multiplication in non-RBC blood cells. It goes to either endothelial wall or other reticuloepithelial endothelial cells like bone marrow, spleen. Skeletal muscles are very common, and even heart muscles are very common, and a lot of times in brain also. And that's when that's where they multiply, and then they again get into these erythrocytic merogony. Those merons will come, they infect RBCs, and then they get into their gametocytic phase. So that's what happens. And what happens is there is this abortive development. So a lot of times hemoproteus can get injected into a bird which is not its regular host. So a hemoproteus from a pigeon may accidentally get into a crow. That crow is not a compatible host, but still that parasite manages to go under these exoerythrocytic merogonies. It infects other organs, and it infects and it multiplies in that organ so much. Eventually, those erythrocytic phases will never be able to infect the erythrocytes, but the other organs are damaged in those multiplication cycles that the host dies. So these are called abortive developments, and that has been seen in parrots in zoos in UK, in London and other parts of Europe, where there are hemoproteus from blackbirds, which are the resident birds, are taken up by midges, are being transmitted into these, sometimes it's parrots, sometimes it's penguins, and suddenly there are deaths. There are suddenly 5-6 penguins which are endangered, which are kept in a zoo, died out. Some endangered parrots from Africa or South America or Australia suddenly died. And then when they did the histopathology, because there was nothing in the blood actually, so the blood was negative. But when they did the histopathology of other organs, they saw the evidences of these stages of hemoproteus that probably has killed them. So that is the abortive development that is very important to look at, how these parasites can be potential pathogens if a non-related hosts are come in contact with each other. So the zoos are very, very important places where this can happen, and there are evidences. So it's very important that these zoos need to take care of these aspects of what all endangered species are keeping and how many of them are susceptible to these things. So as I was saying, the absence in PCR. So what we do is we generally take the blood sample from the bird, we do the PCR, and then we see if there is a positive band, and that means there is a malaria infection. But a lot of times, as Farah said, that there is an acute phase when there is parasite is a lot more in the bloodstream, in the peripheral bloodstream, and then there is a dormant phase where it goes into these other organs and stays dormant there until there is enough environmental conditions, there are more mosquitoes, bite rate, mosquitoes or vectors, the bite rate increases. And that's when the parasite again gets activated for its erythrocytic merogony. If that is not happening, and if I sample the blood, the parasitemia, that is number of parasites in the bloodstream, will be so low that my PCR will be negative. So all PCR negative does not mean that the bird is sure shot negative for your parasite. It can still be positive, but it was not in a parasite, it was not in its bloodstream. So that is very important when you're looking at the data of, look at the presence and absence of parasites. The absence may not be true absence every time. And it's not easy, because ideally you need to have the sample of the tissue, and we don't know which tissue that parasite would be residing in, and it's almost unlikely to not harm a bird and take a tissue sample of something like liver or spleen. So birds are very tiny. So that's one big hurdle when it comes to studying malaria in depth. So when it comes to parasites and hosts, I had mentioned it in the other side, but I didn't say it, that there are generalist parasites and there are specialist parasites. So what are generalist parasites are? These are some of these parasites which are able to infect multiple hosts. Those hosts may be closely related to each other, or sometimes they are not related to each other, very phylogenetically different, evolutionarily different hosts. But these parasites are able to infect them and goes through the transmissible cycles also. So they're able to further infect some other species. So these are the parasites that are the generalist ones. They don't have a very tight association to one host, but they have a diversity. There is a host breadth that they have, which is higher. And then there are some parasites which are very specific to one host. You will find one hemoproteus in myna and not in anything else, only in common myna. And even if it goes to any other host, it does not survive. It's a dead end. So these are very specialist hosts. And what we are, we have to be, in terms of conservation or in terms of disease ecology aspect, is the generalist host that can actually switch, that can jump, they have ability to infect different hosts. We have some data on a certain parasite is known to infect 5 birds across the world. That doesn't mean its host breed would be just 5. It's just that we haven't sampled enough. So having that parasite in your system has a risk of that can infect some other bird also. And then there are some host birds which are also very generalist hosts. Yeah, sorry.
I have a basic question. So you said that host switching is usually an evolutionary change. So is there any incidence of factor switching, or so do they have a evolutionary basis or environmental basis?
I'm not sure if they will have a evolutionary basis or environmental, but there are records. There are several papers that gets published regularly where they just sample mosquitoes at large. They look at blood meal as well as the salivary gland. So you can't, if you just sample the mosquito and look at the blood meal, that doesn't mean the mosquito was in a transmissible phase. It may not be competent vector. The parasite has to be in a sporozoid phase in its salivary glands. So to cut the abdomen and only do the PCR or do the smear of the salivary gland. If it was present in sporozoids, then it was ready to transmit. There are some instances where other mosquitoes, including other genera, apart from Culex, have been found to have those sporozoid stages. But I'm not sure if that will have an evolutionary basis or a more ecological chance events. It could be a compatibility, but not an evolutionary history, just by chance it was compatible. We need more and more data to determine that, of course. With mosquito body, it can be up to from 10 to 16 days. And it is very, so yeah, he asked what is the time period that parasite takes inside mosquito's body, right? Or its vector body to be ready to be transmitted. It can be from 10 to 16 days up to almost 20 days also. And that is very much decided by the external factors like temperature seasonality, how fast the parasite will go through its sexual reproduction and other stages and be ready. And I'll cover that later, but that's what determines. So if you have very highly fluctuating temperatures throughout the day or very cold regions or very hot regions, then even if the parasite goes into mosquito, it will not get to a stage where it will transmit.
This sounds more like amplification, no? Than...
I mean the effect?
Yeah, generally, because you have generalists and specialists and reservoirs and...
Right, I mean...
Has it been specifically tested or amplification, dilution?
No, as far as I know, there are multiple evidences. I think the scale at which we look at amplification and dilution is also matters a lot. Even whether I'm looking at a parasite as a community, all Plasmodium together, or all Hemosporidiums together, or I'm looking at just one particular lineage of Plasmodium, it may act differently to different types of situations. It may be amplifying in a different environment, but in some other parts of the world, it may be actually going under dilution. Depends on what host it is acting. So it's quite complex in that sense to comment, but I think Guha has a comment on it.
So I will be talking a bit about this in my research talk. So hold that question.
So something like, mynas or sparrows, yeah.
We have studied some cross-reactivity between these different malarial parasites. We have studied like
cross-reactivity in the sense?
No. I want to ask, like we are studying or studying.
What do you mean by cross-reactivity?
Cross-reactivity means how antigenically similar these parasites are, human or from the bird.
So, right.
Will also detect some other pathogen, but that antibody was not particularly raised for the other.
Right. All I can tell you is the studies are happening. The studies are happening, but they're happening with very few, very regularly found strains or the lineages of parasites. The parasite lineage diversity is so high, there are only few which are very commonly found across the world, and those are the ones that are being studied. I don't have an answer whether they have found an answer or not, or what is the exact answer to this.
In live birds,
studies.
right?
Yes.
You'll have to infect birds and...
No.
Oh, just test, cross-test.
So ELISA-like thing, I think. You look at the antibodies, if they're present or not, right?
But I'm sorry, why are you asking that question?
That is because what reservoirs are different.
I mean, what he has in his mind is, so when you test this serologically, there might be a chance of false positives or false negatives. That is what you have in your mind, right?
Thinking about can I use one of the malarial parasite as a vaccine in human if it is not like causing disease?
Oh, between
and
humans
it is
and birds?
neutralizing.
Are you asking specifically between humans and birds?
Yes.
At least I'm not aware of if there is any.
He has got a broader picture. Like, see, avian parasites, they are not compatible to replicate or cause disease in humans. That is the first assumption that probably you are having. So, if there are any neutralizing antibodies that have been developed across commonly, then probably those specific antigens could be used as a potential vaccines in humans because they will not, probably cause the disease. So that is what he has in his mind when he's asking.
I think I can try to take an informed guess
Yeah,
on this.
sure.
That if the parasite is not compatible, it will not be able to infect the RBC. It will not go under very high parasitemia. So those few parasites that may get injected may just be either taken over by a very, very frontline immunological response from the human body if it's a bird. Yeah, that's also.
Sorry, thanks. Which for a parasite, it's very huge difference because all the kind of nutrients and things which you are getting in, say, for example, birds, they have nucleated and humans do not have
Yes, yes, I forgot to mention that. Birds have nucleated
It really
RBC.
makes a huge difference for parasites, how they develop and how they function, basically. And I think some of those things, I think, are playing a role, how the parasite actually exploiting that environment in order to persist in a host.
So does the human is having a malaria because of this birds is having malaria because is this malaria spread from birds to humans? No, right?
No, not that way.
Not that analogy is not right because some virus infections are spread from there is a transmission between.
But this is
bats.
not the case here.
This is not the case.
No. Yeah. Yes.
Okay, because previously you mentioned about the rodents and the mammals were study models for the before birds were preferred, right? That's why I
So
told.
the evolutionary jump of malaria plasmodium from its ancestral plasmodia that was there, and some of them were infecting birds, and the evolutionary jump into mammals and some of these specific mammals is fairly recent, and that is a very clear shift. I mean, there is a clear evolutionary divergence between these two. So at a current scale, at a contemporary scale, these two are not compatible and there is no zoonosis happening. There is no transfer between these two. Whatever has happened evolutionarily millions of years ago.
Okay.
And has evolved, then co-evolved with humans or other mammals separately.
Did you study any of the poultry things while studying this malaria?
No, I haven't studied poultry things, but
because the poultry things are the major affecting area, right?
Yes. Poultries are getting affected. There are a lot of papers you'll find from across the world on poultry.
Yeah, and chickens. Okay. Oh, okay. Because we are much more very keen, very close to these chickens rather than these wild birds. So we are actually the one of the consumable thing. No, not the phylogenetically, because we are the interaction thing. Because the interaction between the poultry things is much more closer than these things. We do consumable thing, right? No, not like that. No, not like that. No.
Yeah. But you're asking about the close proximity, right? Yes.
Proximity.
But again, as I said, I mean, even if the modeling will be different from malaria because even if there is a close proximity, it's frequency dependent.
Okay.
As no matter how close I am to another host, it won't transmit. And there is an evolutionary divergence. Even if it tries to transmit, it won't succeed. I'm a dead-end host. I'm not even allowing, I mean, a human is not allowing a bird malaria to even grow in any case in its body.
Okay, yes.
I'll give you an example. So there is a Plasmodium gallinaceae, which is very specific to chickens. It's found in poultry. When poultry have malaria outbreak, it's mainly because of Plasmodium gallinaceae, and it was first time isolated from India. It's very difficult to find in chickens in the normal poultry otherwise. So what happens around the globe, a lot of labs, when they start the work, they get the Plasmodium gallinaceum as a positive control because you need a positive control to do PCRs. The time when you're doing your PCRs, and if you start getting gallinaceum in your wild bird, which means you have a good contamination going on in your lab. Because it's just not, you cannot find it in Gallinaceum, very rare. I mean, I think Japan study one showed, and we proved it was the contamination because of that. So.
Okay. And some of these birds, I mean, if you look at the birds that are, known to be harboring multiple lineage of parasites which are not related and tend to act like some kind of reservoirs. I mean, they are the birds that are allowing multiple non-related lineage of Plasmodium or Hemoproteus in different regions, not single bird, but as a species. There is a clear this thing that, a lot of sparrows or finches, mynas and blackbirds, thrushes are always the ones that you will find are getting, a lot of infections. Even in a patch of forest, I'm sampling different birds, and I get an array of parasites from this region. A lot of them would be infecting blackbirds or finches or mynas if they're available. So that is something also happening. And so look at this. This is a simple case from North America. A simple study. They're looking at different parasites and different birds, and then this is called tanglegram. You have a host phylogeny. These are all birds that are sampled and how much evolutionary related they are. And then you have a parasite phylogeny. And then it's simply kind of a match the pairs, what all parasites and whatever your data is, this parasite is found from all these birds, it tries to connect. And if you can see here, this is a Plasmodium, which is actually going to multiple different birds which are not a single lineage of Plasmodium KZ1, has infected multiple bird species which are not evolutionarily related to each other. So this clearly probably is a generalist Plasmodium in this region for sure. It is able to infect multiple birds. It is able to get transmitted between them. And then you have something like this Melospiza, which is type of a finch, type of a sparrow, which is receiving infection. I mean, it is getting infected by multiple parasites which are not related to each other. So you see this kind of generalist host which is able to harbor different types of parasites in them. And then you have something like this, the parasite that is able to transmit and infect multiple hosts. And when these two come together, yeah.
So what kind of traits make that generalist so successful?
This one? I mean, what type
traits in parasite, parasite.
In the parasites?
Even host, I mean, mostly parasite.
What type of traits that allow them? I'm not sure if that is very well known. I mean, we have to look at different molecular, at molecular level, different regions of adaptations of genome that could be allowing these parasites, certain parasites, to be able to infect multiple hosts than the other.
Can I just jump?
Yes.
So for example, how would you investigate that? So for example, if you would have to do something like comparative phylogenomics of these different, pathogens, and then maybe test for selection on specific lineages to see if there's more, either there are specific changes in specific genes which, allow one to be, or I don't know, you can't say allow one, but it's like strong divergence through selection in the lineage of the successful pathogen. That's how you would test for it. Does that make sense? Like, is it very different? And if it is very different, are the differences something which actually change it in some way, change the proteins in some way, change it in some way?
And those changing regions, are they common in all these lineages which are acting like this? Then you will have some pattern, and then you can go for processes. So when this...
Sorry, and in terms of the hosts, that's something which we'll get to in the afternoon more. Like, what is it about the hosts? Yeah.
So when these two things come together, in terms of, these kind of, you have a parasite that is able to infect multiple lineages, and then you have certain hosts that are actually able to get it and transmit it further. That's what happened in Hawaii. So Hawaiian Islands are completely isolated. They formed volcanically, so there was not like a landmass that shifted. So they evolved, so they formed volcanically, they emerged out of ocean, and then the birds colonized revolutionarily on these islands. And then they evolved there, and there were very specific, very highly endemic lineages of species of Hawaiian honeycreepers. Several species were present in Hawaiian Islands, and somewhere around 1830s, the first time a Culex mosquito was taken, where the colonizers were going all around. They were having those big ships, and mosquitoes could easily travel, hitchhike on those, And there was no mosquito on these islands because by chance the mosquitoes never reached these islands. These islands were colonized at multiple times by multiple different taxa. Mosquitoes never reached. Biting midges never reached. So the birds on these islands evolved in the absence of malaria. Now imagine, I was talking about the evolutionary arms race. You take out one from the race, the parasite on the other hand, perhaps on the mainland where it was infecting other hosts, kept co-evolving. But these birds completely stayed out of race, and then they were out of practice, simply speaking. And what happened is when this the Culex mosquito was introduced, these people also carried a lot of pet birds and a lot of other birds from India, from other parts of the South Asia. A lot of birds were taken as pet birds, or just to have some familiarity, and they were accidentally or intentionally released. Something like red-whiskered bulbul, common maina, shama, Indian shama, white-ram shama are very commonly released birds on many of these islands which are non-native. And when you have a sparrow or a maina, which there is a high amount of parasite prevalence in them. That means a lot of birds, out of 100 birds, a lot of sparrows are infected. So there is a good chance of infected sparrow getting there. And then you have a Culex quincifasciatus, which is a known vector, a very southern house mosquito, a very common mosquito, and is known competent vector of avian plasmodium. And you have a parasite called Plasmodium relictum, which is able to infect multiple hosts which are non-related. All these things came together and got spread onto Hawaiian Islands and started infecting the Hawaiian honey creepers. And within few decades, several species went completely extinct. I mean, there was no time. Now that the parasite is so ahead in the race that the birds just could not compete well, and they just died off. There were mass mortalities. What happened interestingly is many of these islands have huge mountains that going up to almost 4,000 meter elevation. And because the mosquitoes cannot go that high up, they are restricted to certain elevation range. And even if they are present, the parasite within them cannot develop because the temperatures are very cold. So a lot of birds on these higher elevations stayed, they survived. But the extinctions all of them were from the lowlands of these islands. And that's what happened. There are up to 90% reductions in population and local extinctions in a lot of these species. And currently, with these lot of hot years coming in, suddenly very warm year is there, these mosquitoes are slowly going up and the temperatures are good enough for them to transmit. And now in Hawaiian islands, little at higher temperatures, some of these species are restricted to certain elevational bands, like Umesh was telling yesterday. These birds don't come down, they're there only. And now they're getting infected because the temperature is perfect. And now they're showing some die-offs. And this is a very alarming situation because as it will go higher and higher, as the temperature will go warmer and warmer, the mosquitoes may go higher and higher, and that will have an even more disastrous situation. But at the same time, what happened is there is something called as Hawaiian amakihi, that is Chlorodrepanus virens, a species of Hawaiian honey creeper that actually went through a mass mortality. But suddenly those species, individuals started again popping up. People have been noticing that those populations are again growing. And then there were studies that happened showing that these birds have gone through kind of a selection drive, and now there are individuals which are able to tolerate the infection. They have somehow managed to kind of keep up with those parasites. Some individuals that had those adaptations, some kind of traits that allowed them to tolerate the parasites, and those are the birds that survived, they did not die off, and they bred. And now that population is spreading in the low elevations, but these birds are carrying those parasites still within them. So we have a survivor, but that survivor is still having that parasite. So, if now this bird decides to go up, infect individuals, you don't even need introduced species, but this is now able to transmit easily. So you have a very interesting evolutionary process happening in front of our eyes, but yet the situation is quite alarming. So these are some of these individuals that they found that, at different islands, these are serological and PCR based, and the number of individuals infected, that is prevalence, is fairly high everywhere. And that was not found in other birds. The other birds are still dying off. They are not surviving. But this particular Hawaiian amakihi has jumped back.
I mean, by chance, did they check how it was happening, why it was happening? At the purely from the immunological perspective, or not from the
immunological perspective?
Yeah.
Okay, I don't know the exact answer for this.
Because there is this one word that you have put, tolerance.
Yes.
So usually tolerance comes when antigen is, in a very high load, and it fails to, actually do the damage. So, I mean, because of that one word, I asked you whether there is something.
So tolerance is an observed tolerance, as per my knowledge. I am not, I mean, I can help you find the answer if there is an answer.
Yeah.
Not a very simple answer that I've come across very quickly in the literature that if they have done studies.
I mean, this is a very interesting case,
Right.
That is,
But this is,
if you could find why and how it evolved.
Right. Definitely, that's why this case is very interesting, because that will allow us to understand that there are some genes, perhaps there are some mechanisms that are allowing, not letting a species go extinct. Why is it that absent in other birds while all of them evolved together? Why one of them could do it but others did not?
That's right.
But where I'm using tolerance, it's an observed tolerance.
Okay, fine.
Right? Because the birds are surviving.
Some clade or something, is there any phylogenetic?
It's an adaptive radiation, they're all related.
So they're all related. So it's not like,
yeah.
That's really odd.
Exactly.
Yeah, but I'm just saying that since they are all related, it's kind of odd that there's different tolerances.
Why not? Can't we just assume that there is already there existing variation? Some individuals have certain level of resistance, or the pathogens do less harm in those individuals.
No, but
can't we?
I think what I would have thought is that, for example, genes involved in immunity and stuff are generally very highly conserved. So I would imagine that given an adaptive radiation, which is generally fast, probably, I don't know, but maybe I'm wrong. No, I agree with your point that individuals may have differences in immunity, but the fact that there are species-specific differences, although they have a recent common ancestor, I don't know. For me, it seems a bit surprising.
So I think your point is very much valid when compared to that tolerance. So the tolerance may be due to the physiological changes also, the effect climatic conditions and the genetic variations also. The species to species, they vary. It will come back with that particular disease or the prevalence. So I can agree with her point. It may be due to that genetic variation. I think it is much more valid because the immunological response.
Blame everything on genetic variation. It's
because genetic variations.
It's like a good black box. Like genetic variation. Something
Because
to do
the
with
genetic variation is the root to every adaptations, right? Yeah.
There are certain alleles do different well. So it's not a black box.
Yeah. So is that known for avian malaria? Protective alleles?
Most of the times they look at these MHC and tail-like receptors, the common mass histocompatibility complex, the immunological response.
But the problem is that MHC is very difficult to study because I don't know about birds, but it has a lot of repetitive elements, and to understand diversity at MHC is really hard. But there's lots of new immunogenic loci people are looking at, like TLRs and so on. I don't know about birds.
They have found correlations, but they haven't been able to clearly say that this is the process that is allowing it.
So for example, with SIV, right? There's a very nice example in chimpanzees where they've shown increase of a specific allele post-SIV infection.
Right.
So something like that, especially with these birds, because so many died, such high mortality, it would have been, maybe with the museum specimens, one can actually target a question like that.
True. No problem. I'll go fast further.
As far as immunology, from the perspective of immunology, where, as you said, MHC are toll-like receptors. This tolerance need not be related to antibody all the time. Sometimes it may related to the innate immunity, where it is nowhere it is connected to antibody. So modifications of this the site of infection, modification or alteration in the period of time, because of continuous interaction with the parasite, that of the host. So it also tend to adapt itself to survive. So therefore, it may change its target site in such a way that it won't allow the parasite to cause the disease and lead into the death of that host. Right. So it supports for the coexistence.
Yes.
And so it always sees that that host survives. Right. In spite of that, it also supports for its the parasite to present there without much damage. So here we can think of innate immunity as Madam said, MHC, which is very, very crucial role in making the host susceptible or resistance. Whatever the host which is quickly responds to that parasite. by altering the MHC. That host will survive. Whatever the host which is very delaying that during that process, which will collapse.
Right.
That is the evolutionary link to the
Very
immune
much
response.
possible.
Thank you.
Yeah, I'm really glad these points came up during this question. In a couple of days' time, we have a detailed session about host immunity. Looking forward to that.
Yeah, I'm looking forward to learn from it. I don't have answers directly. But right now we have a pattern and we don't know about the process, that's for sure. So, yeah, but I mean, yeah, sorry.
I'm just, I don't know, I just want to know what MHC is.
Okay.
Because, I mean...
I know the full form. It's major histocompatibility complex. Yeah.
Those are the proteins. I mean, we have got two, MHC1 and MHC2. MHC1 is expressed across all the nucleated cells. Each and every cell, you have got MHC1. But MHC2 is one that is present on antigen presenting cells. So these help, I'll not go much deeper. It is just that these will help to differentiate your own cells and antigens. So that is what broadly MHCs are.
Thanks. So this is just a quick representation of how the elevation has a clear response to the disease, in the sense that this is responding to elevation. The birds at high elevation have very low occurrences of diseases, and but very recently, so a lot of Hawaiian work happened on the smear-based technique. So I will get to that, but most of the times earlier, when PCRs were not so frequent, not so cheap, it was very costly. Best way was to just take a blood drop on the slide, do the smear, fix it, and look at under microscope. But that may have actually, some infections may have evaded the eyes of the investigators, because if there is a low parasitemia, you may show prevalence as 0. The presence absence 0, 1, there will be a 0, but there could be 1. The occupancy model from yesterday, you can't know. But when they did the PCR and qPCR is a quantitative PCR which will actually tell you how much parasitemia is there, and even the smaller infections will be picked up by very high sensitive PCR. They realized that some of these birds which were claimed to be having very less or no infection or very less prevalence are actually having fairly high prevalence. If you look at the adjusted prevalence values, the red ones are what they found from the qPCRs in Hawaiian birds, and the blue ones are from the original prevalences that were noted using the smear-based techniques. And we may have to rethink about some of these birds, like for example, Japanese white-eye, which is an introduced bird on Hawaii, was thought to be resistant to malaria because very few instances that was being found infected. But look at this. So this is in white-eye, Japanese white-eye. Look at the difference. There is a difference, some amount of. So we have missed some infections. So the technique that you used to investigate is very important, because your prevalence measures may differ and your all conclusions can differ based on that, right? Now that comes to the third part, the factors affecting the parasite prevalence. What exactly affects prevalence? And most of these that I'm going to show you are correlations. People have done some studies. They have found that, okay, certain parasites are highly prevalent in certain birds in certain regions, and there are some environmental factors that are correlating with them, and we don't know the processes. And is it really parasite responding to some disturbance, some forest type, or is something else happening? And it's worth thinking about. But there are some of the patterns that are very interesting to look at. So we have habitat at the highest scale at the top. We have certain forests that are natural forests that are not yet disturbed so much, and then we have certain which are urban forests or disturbed forests. There is a lot of human encroachment is there. Then we have birds as hosts, since we're talking about avian malaria, and these birds are responding to this habitat. So there is something called as if they have very high disturbance, then a lot of common species tend to stay in high numbers, but a lot of rare species tend to just go away because the disturbance, there are some very micro niches that they're looking for are gone. So we have a kind of a imbalance in the diversity. You have few species which are high in number than the others. So that happens. It's called biotic homogenization that can happen due to the disturbance that is imposed in certain aspects. And you have a natural forest where you have diversity is fairly, in its natural form, where you still have some common species which are in high numbers and some species are in low numbers, but you have a lot of rarer species. The overall species richness also goes high, the diversity also goes slightly higher. And then you have vectors that will also respond to the hosts available, Even sometimes there are vectors that are looking for certain host. For example, in an island in New Zealand, they managed to eradicate one of the invasive rats, and one mosquito population went completely extinct. The 2 years of continuous survey, they didn't find a single mosquito, while the other mosquitoes stayed back. So this mosquito was specifically targeting rats and nothing else. And they took out one, the other thing went off. So this is another big black box. We don't know how mosquito preferences are. And are they really biting birds? Just because there are mosquitoes doesn't mean there might be malaria transmission. That is very important. But it's still important to see how the mosquitoes or vectors are responding to host and to habitat. Because they are not directly only dependent on host, but also where they breed, how much water is there, what kind of structure is there. And then you have parasites which are dependent on these, and which in turn dependent on these. So look at how complex this system is. And then you have certain Plasmodium and Hemoproteus. Plasmodium acts differently, Hemoproteus acts differently. We have more commonly occurring Plasmodium lineages that are known to be generalists, infecting multiple hosts, while in a broad sense, a lot of Hemoproteus species are very specific to their host. Very few times you see a Hemoproteus infecting multiple birds. So we have within within parasites also there is a trait difference at a broader scale, and each lineage might be different. Within birds there is a trait difference. Some birds can harbor more parasites, some cannot. So this is the system that people have been trying to look at and understand what could be happening in the wild. So this is a very nice paper by R.N.M. Sehgal, where it has looked at a good literature review of how the different parasites are responding to the habitat parameters. So there are several studies that are nicely accumulated together, and I will share this in the drive. It's a very, very well written review that you should go through. And it looks at, what are the landscape features that are commonly occurring, people have tested, are known to show correlation with parasite prevalence. So the vegetation, elevation, as we already know, it's a direct impact. Then there is disturbance in terms of urbanization or plantation. You may have still greenery, but it may not be forest, it's just monoculture plantation. That may impact very differently. Then there are climate features like temperature, rainfall, seasonality. Seasonality will directly affect the vector competence to develop that parasite inside its body. And then you have host community, where you have how much diverse the hosts are. Remember the first day Uma talked about dilution effect and the amplification effect. The host diversity will impact parasite prevalence. Now we are not talking about parasite diversity, but prevalence. And the phylogenetic relatedness of the hosts. If you have more closely related hosts, it's possible that some of the parasite might also be closely related because there is very one-to-one host association. So how much of a phylogenetic diversity you have can also tell how much diversity of parasites you have, and the prevalence may get impacted by that. And then there are individual species-based these things, that what is the body size of the species. A waterfowl or a jungle fowl or a large size species will have more surface area for vectors to come and bite, as compared to smaller species which has very little. Most of the times the mosquitoes manage to bite at the eye. So imagine a very small 4-gram bird which has very tiny eye, less likely of getting bitten by mosquito. Even the tarsus are very small. So large bodied birds tend to have more infection levels because they are able to transmit. And the specificity of each species, that is evolutionary or ecologically maintained. So some of the landscape features that are known are these are from the studies, and very interestingly, the distance to river or water body is negatively infecting Plasmodium prevalence. You go away from the water body, there is less prevalence of Plasmodium, probably because of mosquitoes. But since it's river or large water body, I think Farah told me long ago that if it's a larger water body, the fishes tend to eat larvae. So it's larger water body may not impact mosquito. It's something worth looking at. And then distance to woodland edge, how far you're going from a woodland edge and into either inside woodland or away, and that will impact. So this is negatively impacting. If you're inside woodland or close to the edge, you have more Plasmodium. You're going away from it, you have less Plasmodium. Then intact forest has a positive correlation. Intact forest in Cameroon. These studies are very specific to certain regions, like Ghana, the forest disturbance is negatively impacting. There is more disturbance in forest, the Plasmodium goes down. That means the undisturbed forest has more Plasmodium, right? In a general ecological studies, we always tend to say that, oh, the disturbance is bad. And we don't know what Plasmodium exactly is going up or down. Is it really pathogenic, or is it a regular, it's an evolutionary adapted a host parasite relation? And then there is, again, these pristine forests used in the study. These days people don't call pristine, nothing is pristine, But these forests tend to have positive relationship on Plasmodium. Oil palms tend to have negative relationship with Plasmodium. And Hemoproteus, there are very few studies that they have been finding Hemoproteus is directly affected by a habitat character. A lot of studies are actually showing very or less or no relation to any kind of landscape character. In Africa, the forest disturbance was negatively correlated, and intact forest is positively correlated. And that's the overall pattern. Again, you have intact forest, you have more Plasmodium, which is very counterintuitive to how the ecologists think, which was a big eye opener. So, oh, I didn't know there was an animation. So this is how it looked like. Look at this. So the brown one is the pristine forest, and this is a fragmented forest, and the white one is young palm oil plantation, a monoculture. And look at Plasmodium, as it goes from pristine forest to monoculture, it's going down. But Hemoproteus is not showing much difference. A lot of you learned about the standard errors and how to look at it. There is huge overlap. There is no significant difference between them. And there are some other things like, in Mexican Sky Islands, they looked at urban forests have higher Plasmodium. This is different from what they were finding in African forests. And even in Atlantic forests, there is higher Plasmodium prevalence in disturbed forests. When there is disturbance, there is high Plasmodium, which is opposite to what they're finding in Africa. So one parasite does not have a single response to a habitat character. It's very case specific, it's very case sensitive. Where you are going, it is differing. So, and then climate features. Again, Plasmodium is very positively correlated with temperature. You have higher temperatures, you have Plasmodium going up because mosquitoes tend to again stay well. They're able to harbor those parasites.
So this whatever parameters we are correlating with the vector prevalence. So we cannot. What I feel like it is like a recipe. That combination should be present to have that prevalence, but maybe somewhere else, something else will complement that thing for its.
Right.
So we may predict something wrong.
Exactly. So what habitat characters you're looking at? Are you looking at all ingredients in a recipe? Most of these studies are not including vectors. There's no idea how vectors are responding. Just looked at birds from disturbed forests, birds from undisturbed forests, and they found a relation. Okay, the Plasmodium is acting differently, but do we know how mosquitoes are there? That is not tested in many of these studies. So that's a big black hole again there. And we have no clue what's happening.
How many? Sorry, what's the black hole? Sorry.
The mosquito part or the vectors are not tested. I mean, there is no data on vectors. It's just a host, parasite prevalence in them, and disturbance and undisturbed forest.
Because it's very difficult to study vectors.
Right. It's a lot of cost and logistical this thing.
Participants are also running around now. That's good. Not only Ansil.
Just have two questions. One is that, so with climate change and increasing temperature, you're saying the Plasmodium prevalence is going higher. So, I mean, I don't know, to some extent the rainfall might also add into my question. Maybe, is it humidity? Does humidity also play a big role?
Humidity would play a big role, but it's more like the water availability for mosquitoes. I'm not sure if the immunity and in terms of its own direct effect, the humidity of the air will impact directly. But if that immunity is maintaining the small puddles for mosquitoes to breed, then they will look for more blood meals. In turn, will transmit more parasites. So,
because what I would assume is like in a disturbed forest, which like if you're talking about like the forest that Umesh was talking about yesterday, where there's active logging and all, you might have like higher temperature maybe, but like humidity may be less because the higher temperature might take away reduce the humidity.
Yeah.
But also at the same time, are there any studies done on like tropical rubber plantations? Because I'm just assuming there's always a lot of water retention in the rubber, where they collect the rubber sap.
There should be. I have to search. I will find it out. I must be there in my Zotero.
Thank you.
Yeah.
Just a interesting study that I'll just share. I forgot the name of the author, but then this wasn't specific to avian malaria, but human malaria. The prevalence of Plasmodium was indirectly connected. This was done in the Northeast to presence of elephants and elephant footprints kind of creating puddles. And I just thought, that was something interesting that they had studied. But yeah, it would be, I mean, just shows how many other factors you could add, variables that are out there.
I can quickly relate to the Tarai landscape where it was a hub for malaria earlier and all the big herbivores were there, elephants, rhinos. Could be maintaining it, who knows. But there is also a river and others. But yeah, thanks. It's very interesting. So this is how the climate responds. Again, hemoproteus responds differently. Hemoproteus seems to be, found more in colder climates than hotter climates. It again depends on how midges are responding, and that's another big black box. We don't know how different midges preferences are, how they're really responding to these environmental factors. Very tiny mosquitoes are still easy to sample. Midges are very hard to sample, even hard to differentiate a biting midge from a similar looking other dipteran on field unless you're a very trained eye. And yeah, I tried, but I failed. And then rainfall, it's uncertain. I mean, a lot of studies have uncertainty around it, that how rainfall would affect the hemoproteus. And there is this study from New Guinea where they looked at, again, understory versus canopy and how different parasites are responding differently. So here, look at this. In hemoproteus, it is more in canopy nesting, canopy dwelling birds than the understory birds. But Plasmodium is more in understory birds than in canopy dwelling birds. It's possible that midges are able to, generally in the buildings also, we tend to get a higher floor because we know mosquitoes won't come that high up. If that is a pattern that mosquitoes not going very high up, then the Plasmodium will be less in the birds that tend to stay in very higher stories in the forest. But midges may be able to go. So that could be one relation. And there is species-specific responses. So these are general responses that you are finding. But look at different species responding to NDVI, that is the amount of green cover, the distance to roads and minimum temperature. Every species has a different type of response. So remember, we were talking about random effect 2 days back and yesterday also. So this is kind of a random effect. If I'm looking at all birds as birds and I have a pattern, different species might be responding differently, and that's a random effect that I may have could have neglected. But I can use it in my model, species identity as a random effect, because with different species, the slopes are differing. The y-intercepts might be differing. Different species may have different thresholds at the baseline of that variable that I'm testing. So that's very something interesting that you can correlate what you have just recently got familiar to as a random effect. It's very important to when you have a mixed species community, host community looking at species identity itself can become a very important random effect, how species are responding. And then we have this dilution and amplification effect, how host diversity impacts parasite abundance or parasite prevalence in turn also. So what it suggests, the dilution effect suggests that we have higher diversity, then for a particular pathogen, a particular parasite, to find its ideal host, some other host may come in between. In terms of malaria, a vector may bite a compatible bird, take the parasite, but the vector is not looking for other same species of that bird. I mean, it takes it from Maina, it's not looking for Maina, it's just looking at another bird, and by chance it comes across Bulbul, it bites it, and there was a dead end. So transmission could not happen. So higher diversity will dilute the contact rates, and eventually the prevalence will go down. In the other hand, if you have diversity that is low, as I said that with disturbance increasing, there is biotic homogenization, very few species, very high in number, then the prevalences may go high. So dilution effect can be interpreted in the other sense that if you have more disturbance or less diversity, your prevalence and parasite abundance may go high. But there is a competing hypothesis called amplification effect, which says no, if you have higher host diversity, you also have a high chance that the parasite diversity may also be higher. For every host, there may be one specific parasite. And if all of them are in good number, so diversity is not just species richness, right? They're also in their own relative abundances. And all of them are in fairly good abundance, the parasite actually might be able to sustain itself in the system. So you may have intact forests which have a higher diversity and very high parasite presence, overall prevalence, diversity. And perhaps that's where the spillovers might be happening. We go more closer to intact forests which has a lot of reservoirs hiding there. Good amount of parasite prevalence also. There is more chance of that contact getting spilled over, right? And then there is correlation with vectors. Very few studies, but what you have to look at is, if you're really interested in vector biology, then what is the vector density? What are the biting rates? What host these vectors are preferring? Are they really only biting humans or only biting specific birds? Or they may be the southern house mosquito, the known vector for Plasmodium relictum, is also spreading a lot of disease in humans. It's biting everything. So the chances are different. and then the coefficient of transmission. There are other parameters that you can look at. And then the habitat characteristic will determine how the vectors are acting and how their abundances will grow. Yeah. So it's a very interesting study that Farah was also involved in, where what they looked at is, I think somebody asked how much time the parasite takes in vector and how it is dependent. There's something called as parasite development rate inside vector's body. So when a plasmodium enters a mosquito, then how quickly it will go under its sexual reproduction, gets absorbed by into its body and able to produce porocytes, and then they are able to reach to the salivary glands and get accumulated there. That time will be dependent on how high the temperatures are, how much fluctuating temperatures within the day are. If I have very low temperature in the morning and very high temperature in the afternoon, then that will reduce its development rate. If I have growing temperatures somewhere around 20 to 25, I am a very high activity, peak activity. But if I have very high temperatures, that is almost 35, 38 degrees, then the parasite activity completely shuts down for a lot of these parasites. And different parasite lineages like Plasmodium relictum, Vivax, falciparum will respond differently to these environmental factors inside a vector, and that needs to be modeled. And also the intraday variation or interday variation. And the reciprocal of this parasite development rate is called as the extrinsic incubation period. That is number of days it would take for the parasite to grow. And these vectors have very small life cycles. Some of these vectors don't live for more than 50 days. So if you have modeled in extrinsic incubation period, the number of days is higher than its life cycle, the vector will die naturally before the parasite is ready to transmit. And this you can easily do once you have models and parameters for different parasites ready. And there is a complex statistic that goes into that you can personally explore. You can actually map based on the satellite-based temperature data. What are the places more likely to have transmissions occurring? What are the places where the temperatures are going in such a way that very less likely the transmissions are happening? So we can actually focus some of the conservation efforts where the temperatures naturally are going up and down and very high likely of transmission happening, and then you can have control operations. So those early detection and control operations can come handy if you have these parameters well tested. I think we covered this. The Plasmodium being more generalist and Hemoproteus being more specialist, and then there is something called as host breadth. So I think that is little ahead. No. Yeah, I think we covered this. So there is nothing different in that. And then coming to the Western Ghats, the case study. I think we have half an hour. And the case study was in Western Ghats. So what led us to think of that? I mean, there was one more student who was working on the Western Ghats birds and the Plasmodium and Hemoproteus within them. And she completed a PhD. She had some papers out, and there was some data available. And what I think Robin explained, what are the sky islands? What are these mountain climatically isolated peaks? And we have these Nilgiris and Palni Annamalai hills. And we have very different habitat, fairly cold temperatures, kind of a temperate mimicking system in a tropical latitudes, which is very interesting and very important to consider. And we have these endemic birds, just like Hawaii, right? But these are not so isolated. These were not, came long ago and then just stayed in isolation. There are climatic cycles that were influencing these birds. They might be extending their distributions. There might be range shifts in the history. And these birds may have different levels of, competence with the parasites that are surrounding them. And what's really happening? Are these birds really in danger or not? And then there are landscapes that are being changed. Look at these Palni Annamalai hills. The shades of red are where your human interventions are happening. There are plantations, there are urban settlements, there are tea plantations, a lot of things are happening. And the greens ones are the Shola grassland and Shola forests. So imagine last 40 years, the rate has gone up. That means a lot of disturbance has been imposed onto natural forests in these areas. And we looked at how disturbance might be affecting the diversity of birds, and is there dilution effect, is there amplification effect, what could be happening. So some of the earlier reports, this was more of an exploratory, let's look at all the 4 sky islands. And this is what Pooja worked on before I started my PhD. And there was a very nice, interesting pattern that some of these birds, like these chilapans or laughing thrushes, have fairly low Plasmodium but very high Hemoproteus. Then there are these, again, Sholakili also have very high Hemoproteus but very less occurrence of Plasmodium. And then you have certain birds like Nilgiri flycatcher where there is no Hemoproteus but there is 4% of Plasmodium. Interesting. Then there is blackbird, 29, 30% of Plasmodium is there in all the sampled blackbirds. Again, a thrush species, which is able to, in general, potential reservoir perhaps. And other thing was that they found that some of these parasites, especially Hemoproteus, are actually diverged across sky islands, 2 different mountain peaks. Sorry, Goha, I'm dwelling into your slides. Yeah. And what's happening that, imagine the host has diverged for millions of years. Okay, so there's some 5 million years difference, divergence between Chilappan or Sholakili between 2 sky islands. And there is a specific parasite that is harboring in that host, a Hemoproteus, which is very host specific. And there is a huge gradient of climate for any vector to perhaps transfer. We don't know about vectors yet. And the FST value, so this is something, a parameter that you may learn further, but that just shows, simply speaking, if you have high F value, that means there is a good divergence between 2 populations, they are not meeting. There is no genetic connectivity between 2 populations. If you have low F value, the connectivity is fairly high. And they found that the connectivity was fairly low. So fairly high. So imagine the host hasn't met each other. There is white-bellied Sholakili, there is Nilgiri Sholakili. For 5 million years, the host hasn't met each other. There is no genetic connectivity in between them. But the parasite lineage has managed to stay same. And how is it possible? I mean, are the random mutations so small or so slow that, it just hasn't diverged yet? It is because physiology perhaps might be same. Or is it that somehow this parasite managed to, cross the gap and go to even 1 or 2 individuals is enough to have, gene flow even once in few decades. So this was a big mystery. But another interesting that came up is, look at Hemoproteus. There is something called as phylogenetic clustering and overdispersion. That means in a given area, whatever organisms are looking at, are organisms in the same area more closely related to each other? Or are the organisms in the same area are distantly related to each other, and each of them are closely related to organisms found in another area? So there can be dry zone species from multiple areas which have emerged from single ancestor. So you will find them phylogenetically close but actually geographically well placed. Or you can have something like Hawaiian honey creepers that all came from one single ancestor. That means same area, all birds which are showing different choices of habitat and other things, but all clustered together. In parasites, they found that Hemoproteus is phylogenetically clustered. That means a lot of Hemoproteus lineages found from sky islands were closely related to each other. That means perhaps Hemoproteus may have evolved independently in sky islands with the community that was evolving in that area. While Plasmodium is showing relatively, on an over dispersion scale, where Plasmodium lineages that are found in these sky islands are actually distantly related to each other. So that can be on more ecological scale, that these Plasmodium communities or assemblages have gathered in the more recent times with the kind of hosts that are colonizing these forests. And then again here also, look at these parasites. Some of these Plasmodium are infecting many birds. This was again Pooja's data. And then these are Turdus merula, that is old name for the Indian blackbird. Another thrush which is getting infected by multiple distantly related Plasmodium. Similar the case in also Hemoproteus, but lot of very close associations can be found in Hemoproteus than Plasmodium. So there is some amount of Hemoproteus being more specialist and Plasmodium being more generalist in these landscapes. And then there is also some amount of evidence that these parasites are responding to the distance from protected area, or how they're responding to overall disturbance and different climatic factors. And there are ways of, you have certain data from certain locations and it is showing you some pattern. You can predict it for rest of the area and have a general pattern that is happening on a geographical scale. And I think Guha will cover more into this. But what it gave an idea that perhaps Plasmodium and Hemoproteus are responding differently to type of habitat they are dwelling in. And it's possible that as you go away from protected area, the Plasmodium diversity is actually increasing, and Plasmodium prevalence might also be increasing. That means you're going away from protected area. That means you're coming closer to human settlements. Disturbed habitats may have higher Plasmodium. So these were some of the patterns that led me to ask some of the questions that I started working on, that are parasites really, are the prevalence of avian malaria, that means number of birds infected by these Hemoprotean sporidian parasites, higher in disturbed forests, urban forests, where there are a lot of human settlements, perhaps that are the ones that are harboring more diversity of mosquitoes, because there are a lot of tourists coming in, a lot of garbage is there, garbage dumps are there. And are these parasites less prevalent in the natural landscape? Because this may have a dilution effect, the other end of dilution effect. You have disturbance, you have less diversity, there will be a boom in the population of the parasites. And it may be more stable in the forests. So this is what a hypothesis is. Natural forests will have overall less prevalence. As you go to urban forests, you will have very high prevalence of these parasites. I have no clue what's happening to the vectors, but I have some idea of what could be happening to the birds. That there could be some amount of homogenization, there will be some amount of diversity changes between these two. I actually, unfortunately, haven't tested it statistically, but that's something that we will look at eventually. And then my quick hypothesis or the prediction was the site, actually it should be, prediction is the ideal term now, that urban forests near settlements should have high infections, while natural forests should have low infections of both parasites, Haemoproteus and Plasmodium. And between host species, I had an idea that, we looked at there are generalist birds and there are specialist birds. So blackbird or orange-red thrush, these are all generalist birds. They are found across elevation, multiple habitat types. And there are endemic birds which are very host specific. So these may have different response. And as the case from Hawaii is well known, that we have generalist bird and generalist parasites, they tend to, they tend to have more transmission and infection levels higher. So it could be possible that Plasmodium is more in habitat generalist birds and Haemoproteus could be more in habitat specialist birds, because there they find a very tight association. And then I came across this hypothesis which said that okay, there's something else also could be happening. It's not necessary, because one of the first bit of sampling we did, we had one site from protected area, one site from an urban area, an urban shola forest, and the prevalence of both Haemoproteus and Plasmodium in that protected area was double than what was found in the urban area. So exactly opposite of what I was thinking. And then that was just one site. Statistically, it wasn't, not a lot of samples, but there was some evidence that made me think that, oh, there could be the other end, there could be amplification. The parasites are actually responding in more amplification effect manner. So the hypothesis I kind of reworked on it. Is it this happening that, you may have natural forest may have higher amount of Haemoproteus, but less amount of Plasmodium, because Plasmodium is generalist. It will favor more generalist birds, and Plasmodium may go more in urban forests. So Plasmodium may have dilution effect, and Haemoproteus may show amplification effect. Both of them can act differently. But I'm not looking at lineages, I'm looking at a broad genus. Specific lineage may also respond differently. So again, the scale at which we are looking at, that may change your hypothesis and that may change your conclusions also. So I asked if the human disturbance is causing homogenization of host diversity. That I haven't tested. This part I haven't tested statistically. I'm assuming there could be, because when you do misnetting, you're not sampling all the birds. You're getting that misnet capture history is not a good indication of what your actual diversity is. But it may have a positive impact on Plasmodium and negative impact on Haemoproteus. So I worked in, we are here, so I worked in Pandianamalai Hills, and I had 6 sites. So earlier I only did these 2 sites, and that showed the amplification, something hint towards amplification. So I did some more sites, and different sites had a different type of setup. So this is, look at this whole settlement, and there is a shola forest in between. This is an undisturbed site, which is the Anamalai Tiger Reserve. This is Pampadam Shola, and then this is again Devikulam, which is very close to Munnar and Devikulam town, and surrounded by mostly plantation. The shola forests are themselves very isolated small patches. And then I have certain sites. I have 3 sites of urban, 3 sites of natural. So there is no random sampling here. There is a well thought that has gone behind this study design. I did not have to random sample. I just needed paired samples. For every urban site, I needed a natural site. So you don't, it's not necessary that every time it has to be random sample. Perhaps if a random sampler was asking for some 60 sites to be sampled or 100 sites, it was not possible for me to cover it in 5 years. So I had to go for few sites but high number of birds. So I managed to total sample 1100 birds from these 6 sites, and they were in paired setup. Each urban site has a natural site nearby. And we catch birds, we take blood samples with misnets. Umesh covered it yesterday. And then also looked at, this is how we have a shola patch. So this is a tiny shola patch which is surrounded by monoculture plantations, some houses, and even if you go further ahead, there is a big township here. So how many nets you place, how you change them, because your capture history will go down. The birds will get used to these nets. They don't come into net any further. So you need a lot of people, lot of nets. They have to be arranged, and there is a gap, in between That was the day elephants came and we all had to run. So we just couldn't sample that day. Same is with Pampadam Shola. Look at this. So with different number of nets and the days increasing, the number of species that is white and number of individuals go up. And there is a saturation. After that, we have to stop. So I could do fairly 200 birds in a 10-day period from these sites. And then what you use for PCR is the investigating pathogens. This is the part which I wanted to focus on. Is there are two types of PCRs. One is multiplex PCR that is developed for hemosporidians, where they have different sets of primers. I think all of you are well versed with PCR, how it works, right? So there are different sets of primers, and each primer is targeting a different region for a different genus. So primer for Plasmodium is targeting some part of mitochondrial DNA. Primer for hemoprotease is targeting a marker in SIDB region, and for leukocytosis is targeting some intron inside some part of, again, the mitochondrial genome, and their amplicon size will be different. So I put all three primers in the same mixture. I run the PCR and I run that sample on the gel, and I get either single band, and I know which size band I'm looking at and what parasite it should be. So hemoprotease should be at 378. Oh, this is not, this is wrong. This is, so hemoprotease is 533 and Plasmodium is 378. This is opposite. So top is hemoprotease. This too means co-infection. A single bird is infected with both Plasmodium and hemoprotease, which is very interesting. And sometimes you get something very at low at this scale, that is 218, where you have leukocytosis also. So that gave me a quick idea of what are the prevalences. I'm not looking at what lineages, but I know how much Plasmodium, how much hemoprotease are there in these birds. But this does not let me ID the individual lineage. Because it's important which parasites are generalist, which parasites are specialists, which lineages are actually infecting multiple birds. That, you know, that tanglegram I need the lineage ID. So this wasn't possible, but at least it gave me some idea of what's happening with the prevalences. And then you have nested PCRs. So what you do, there is a first PCR where a certain primer, you amplify a larger fragment of SIDB within the parasite genome, which is embedded in the blood sample of a bird genome. And then you take the product of that first PCR as a template for second PCR, and that will give increase the chances of amplifying the actual parasite DNA, that actual fragment of 417 and base pair sequence of SIDB. And then there is a huge amount of database available on NCBS, and there is something called as Malawi database that is just opposite of avian malaria, AVMAL Malawi. But that database has all the data on all three hemosporidians, hemoprotease, Plasmodium and leukocytosis. Whoever works at any point at any corner of the world, they submit their sequences to Malawi. And there is a blast tool for that also. You blast it, and if there is a lineage that hits, you know it is the thing. If it doesn't hit, the divergence is one base pair. There is one base pair difference, the lineage is split. Their haplotypes are treated as separate lineages, every haplotype. So it has to be 100% match, otherwise it's a different lineage altogether. And this is how you see it. So, but again, for hemoprotease and Plasmodium, there is only one set of primer that amplifies it. They haven't been able to split it further. So either you can have hemoprotease infection, this could be either hemoprotease or a Plasmodium. Unless I sequence it, I blast it and I see what it is going with, I don't know whether it's hemoprotease or Plasmodium. That's, which one? Ladder? Oh, ladder size, this is 100 base pair ladder. So the amplicon, comes nearly somewhere around 500, but these are along with the ends, so we managed to take out 478 base pair out of it. So this is where this comes handy, because I know for every sample, whether it was actually hemoprotease or Plasmodium, or it was co-infection, and then I can treat it accordingly. And if there is a difference, I was expecting hemoprotease when I got Plasmodium, it could be cross-contamination, it could be a mixed infection that was missed. But it's ideal that you use both the methods to have the actual prevalence data, because this method can miss some of the infections. This infection showing there is no infection for this sample, but nested PCR is more sensitive and it can give infection. So there are some false negatives in both sides. So ideally you put them all together and have a constituted data. And when you have co-infections in your sequence data, you can see double peaks. And you cannot know, how do I know that which goes to C and which, If it's just one case, I can have probabilistic, I can just try one with C, one with T. I split them apart, one sequence with C, one sequence with T. It may go to hemoprotease or Plasmodium or two different lineages of hemoprotease. But when I have 5-6 sites of such double peaks, there is no way I can know that which goes to what, and that is a new lineage or it's a known lineage, because it's just one base pair difference that will change the whole scenario. So most of these sequences have to be discarded, unfortunately.
But
yeah.
Can you sorry, you're trying some other
I
molecular
tried
approaches?
phasing and everything, but in phasing also it will assign, let's say there is a C and T, two peaks, and then there is A and T.
You can't phase it.
Right, I can't
Yeah,
phase
I see
it.
what you're saying, but have you tried what's that thing called? I'll get back to you the name, but is it Tachman type stuff?
No, I haven't tried that.
We can talk about it.
Sure,
Allele-specific PCR.
Okay, definitely. And then, you have these many variety of birds. Look at the sample, the body size. As I said, the host traits matter. You have a bird as big as grey jungle fowl. It's very closely related to our domestic fowl and chicken. But the plasmodium that I found in this was different than the gallinaceum. That is very specific to chicken. This was different. This is a new lineage that we found. Then as small as 4 gram of Nilgiri flower pecker. And you have this much of diversity. And I was trying to look at how to categorize them as host, generalist host and specialist host. Which ones are habitat generalist, which ones are habitat specialist. I could simply do all the endemics are specialist, all the non-endemics are generalist. But is it really that categorical differentiation? Is it really interpreting ecology of those hosts properly? Perhaps not. So I went to this part where I looked at the elevational range of each host and I took the minimum elevation of every host. So it gave me a continuous scale. Instead of categorical, host, specialist and generalist, I got a continuous scale. For every bird, I have what is the minimum elevation. If that minimum elevation is only higher, that bird is elevationally restricted. It's more specialist. If I have elevational, the lower elevation limit is very low, that means the bird is found across. That means it's more generalist. Right? So this is the one that I used.
What if the range is not completely from the bottom to the top? What if the range is really restricted to the bottom? Then
so I sampled only on the top of the mountain. I did not sample the lower part. So if there is any bird which is found only here and does not come up, it never came into my samples. So my samples are restricted to Shola forest. Okay. Yeah. So automatically I only get birds which are either distributed across or only restricted to top. And look at this. This is a general prevalence data, a part of my data. And see the natural forest. So green is not infected birds, and the other colors are infection with other parasites. And what you can see on a general trend, there is still slightly higher infection in natural forests than in urban forests. And so since we have been talking about models a lot, this is how I use my data in a binomial framework. I did not use 1 zeros. Yes. Which one? This one? No, this is just visual interpretation. I'm not statistically inferring on this right now. And this is how I arrange my data. Instead of 1 zeros of every individual, I took proportions for every species, but in different sites. So I have location, that is my each location. I categorize the location as is it an urban forest or natural forest. Then I have species of bird that I have sampled, whether that species is generalist or specialist. But instead of this, I use minimum elevation, which is on the other side, as a continuous scale instead of this. And then total number of individuals of this species from this location, and how many are infected with Plasmodium or Hemoproteus or Leukocytosome. So this is the proportion data that I get. And inherently, the species ID becomes a random effect because it is very well known from all the studies that different species may respond differently to parasite. So species goes as a random effect here. And what I'm looking at is the model is whether the prevalence or the infection probability is correlated with site as either urban or natural, because I want to look at how urban or natural is, how the prevalence is differing between the two. Minimum elevation is the prevalence increases with the minimum elevation of the bird or not. The interaction of both two. That means what? A specialist bird in a disturbed site versus specialist bird in an undisturbed site. Are they two responding differently to the parasite or not? That's an interaction. And this is what you get. I mean, you remember the odds ratios that we talked about. The odds ratios are simply probability of infection versus not infected birds. I mean, the ratio of the odds. So this is the odds of urban forests and versus odds of natural forests. So these are the parameter estimates that you get. And if anything is below 1, that means it has a negative correlation with the specific variable that you have. So that means for Plasmodium, as the minimum elevation increases, as the host becomes more specialist, the prevalence of the Plasmodium goes down. So more specialist birds, elevationally high elevation birds have less Plasmodium. The birds that are found across elevation, they have more Plasmodium. For Hemoproteus, that was not the case. I mean, the standard errors were too high to have any conclusion. It was not showing any relation. But Hemoproteus does show that it is more in natural forest than in urban forest, because here the estimate is on the other side. The odds ratio are more than 1. So the odds of urban forest to natural forest are more than 1. So that means natural forest has higher infection, right? And then there is an interaction. So best way to look at it is this. Look at the Plasmodium prevalence. Whether it's in natural or urban site. Yeah, Ashwin. Sorry.
Go to the previous slide one. This previous one.
Yeah.
So you have mentioned some of the migrant resident birds. Do you come across any of the single species where you found in both in urban and their natural habitat? Same species we have found both in urban and natural habitat.
Yes, there are several species that are found in both urban and natural forests. They're common species.
If there is any migration of that bird, leads to the migration of that diseases.
No, so this migration is the long distance migration. There are warblers coming from Himalayas and Europe that are inside. So I was just trying to look at if the migratory behavior of the bird is having any impact on the data or not. Yeah.
That's why there is a difference between that species of the parasites. So,
I mean, a lot of migratory species have very specific parasite that they are carrying, and it is not found in the local non-migratory resident bird. So they don't transmit, but they do carry it. They probably get it in their breeding range, which is either in the Himalayas or in Europe. Okay. So look at this. Both natural and urban forests, whether it is, the Plasmodium is acting same towards minimum elevation. As your minimum elevation goes up, that means the species becomes specialist, the Plasmodium prevalence goes down. But, so there was no significant result for interaction term here. But as you go to Hemoproteus, there was a significant term of interaction. The two variables are interacting with each other, meaning in urban forests, the Hemoproteus in specialist birds, see, this is minimum elevation. Higher the minimum elevation, specialist the bird. And specialist birds, Hemoproteus is going down in urban forests. So in urban forests, if I look at specialist birds, there is very less Hemoproteus. But in natural sites, it's going up. So Hemoproteus is acting differently in specialist birds between sites, which is very interesting. And why it could be happening? Why these, and Hemoproteus, if it is very host specific, are the hosts responding differently to these natural and urban forests? And that's how the Hemoproteus is also responding to them. It's possible that that is the case, right? This is worth thinking. This is another trianglegram. See, this is the JRW4 Plasmodium relictum, the same that is creating havoc in Hawaii, is found in one of our study sites. It's interesting, it is already infected, it's found to infect pied bushrat and Nilgiri flycatcher. One of them is endemic. There are other lineages, and there are less number of lineages that I found overall. And this is just, look at the blackbird again. Multiple, distantly related lineages are actually infecting blackbird of Plasmodium. Again, this is Hemoproteus in urban forests. A lot of one-to-one relations. Many of them are very one-to-one relations, not much of a divergence on both the sides happening, except for some of these blitzreed warbler, which is the migratory bird, which is actually getting infected by multiple, but fairly closely related parasites, right? Again, for Plasmodium, it's slightly different here. So what?
To that one?
This one?
This, yes, Scimitar babbler is also showing some of the Hemoproteus from different lineages to be.
Pearly laughing thrush, it is the top one. Scimitar babbler does not have, there was a slight error in the data that I could not take care of.
Sorry.
So it's the Pearly laughing thrush that has, that is getting, infection from multiple non-related parasites. Yes, actually we found that. So there are some parasites which are globally distributed, some lineages that are also infecting some flycatchers in Europe and other places. And we found it here, that too in Pearly laughing thrush, which is the endemic bird. So there seems to be some Hemoproteus lineages that are globally distributed, fairly generalist perhaps, and are also acting as generalist. This call2 is infecting a lot of birds, and that is from collared flycatcher from Europe. Very interesting. So what you look at, this is a very visual interpretation, right? There is a quantification in this. So what you generally look at is host breadth. For every parasite, I look at how many hosts it infects. So the simple host breadth is number of hosts a parasite infects. But if those hosts are very phylogenetically similar, a parasite infecting 3 species of pigeons, then then it's still a specialist of pigeons. It may have 3 species, but it is not infecting a woodpecker. But there is phylogenetic host breadth. That means I look at the total phylogenetic distance between all the host, all the host that it is infecting, and I take a summation of all that. If I have more distantly related hosts, then I have higher phylogenetic host breadth. The phylogenetic distance will be higher. If I have a lot of them are closely related, all pigeons, the overall summation will be less. Something little higher is, other way of looking at it is mean phylogenetic distance. Instead of adding it up, I just look at the mean. It is going to act the very similar way, because if I have more distantly related host, it is infecting a pigeon, a woodpecker, a myna, and a sparrow, then it will give me a higher mean host breadth, mean phylogenetic distance of the hosts that it infects. Pairwise, yes, mean pairwise. And there is something called as standardized effect sizes. So what it does is, instead of looking at just phylogenetic distance, mean phylogenetic distance, it puts a null model, a randomness aspect. What if, is it, what I'm observing, is it a part of a random distribution? Am I just by chance seeing some pattern? So what it does, it resamples the data and it will assign this parasite. It knows that parasite infects 3 hosts. I have observed them to be 2 species of pigeon and 1 woodpecker. It's going to now shuffle it across all the birds that I have sampled and randomly assign any random 3 hosts. And that will do 1000 times. So it will get and every time it will calculate a host breadth, mean phylogenetic pairwise distance. And it will have a distribution of that. And the mean of that will be your random mean. By random chance, what is the most likely phylogenetic host breadth I am getting? And then I look at my observed one. And is the observed so far from the random mean? Then what I am getting is something very more than by random chance. So if it is negative, that means my host breadth is less than by random chance. That means that parasite is more specialist than by random chance. If it is plus, that means it's more broader than by random chance. That means the parasite is more generalist than by random chance. So that's how it works. So this is how the generalist will have higher host breadth, same thing. Yeah. And then these are some of the patterns that I'll just quickly, I already showed, these are the specialist birds. Look at this. Kodaikanal is a disturbed site. Then Deviculum munar are disturbed sites. Fairly low infection, 35%, 50%, 0. But again, number of individuals are very less. Look at Pampadam shola, Annamalai tiger reserve, Berricham. These are protected areas. And the infection just shoots up like 66%, 85%, it's very high. See, look at the Sholakili. Most of the disturbed sites, so Kodaikanal, Deviculum, Munnar, there is 0 or 4%. But as you go to an undisturbed site, that is protected area, the infection levels are high, 44% or 38.5%. And again, Berricham was kind of an outlier in this, and we are still trying to statistically test it, how this is. Black and white are also similar manner. So the specialist hosts are actually responding differently to the parasite infection in different types of habitats, right? And then I will look at mosquitoes also. So this is still ongoing work. We have got the data. I'm yet to analyze it, but I'm showing just, I put 1 kilometer, 1 kilometer larger grids, and then I put 100 meter by 100 meter random grids. And in each grid, we put a mosquito trap for one night. We sampled mosquitoes and looked at a gradient from urban center towards forest. Is the mosquito community differing as you go away from it? Same for the Munnar. And then we looked at some of these. So we actually built a 3D printed trap. So the CDC trap that you have to import it from US was fairly costly. So we got few, we looked at the design, we thought why not we build it ourselves, and we managed to get it. And these worked as efficiently as the CDC traps, and just half the cost. And just quick look at the data, what may look like. This is from a very short sample size. What you can look at is, you can look at the mosquito Shannon diversity, or the mosquito species richness, or their abundances, and are they different between disturbed and disturbed. Don't look at the pattern. This is just based on few 5 sites randomly, just to show you what kind of analysis you can do. Or you can do something like NMDS that yesterday Umesh was talking about. You put on a multi-dimensional spectrum, you put your sites and see how the sites are arranged. So instead of a multi-dimensional plot, it's on a lat long. So these are actual locations. But if you just look at this diversity, every color is a different species. So as you go away from center of the town, your species composition changing, there is a beta diversity there. There's a turnover of species happening. So these are some of the plots, these are some of the analysis that you can do. But this is not the final result. So this is not the ideal, this is not the results. So thanks a lot. These are my people who I worked with, supervisors, all 3 of them are here. and they have guided me, and all my team of people who helped me on field and in lab, all of them risked their lives to, to elephants and bears and whatnot. Yeah. So thanks. So these are the nets that were broken by elephants.
Quick question.
So it's a quick question. So I want to ask, like you are saying about urban forest and then the naturally occurring forest.
Yes.
So is that avian malaria is always stick to the forest, or like how about a city like Bangalore? There is no prevalence of avian malaria in Bangalore.
We don't know that. We haven't sampled birds from. There is. Farah has. Yeah. Actually, avian malaria is very ubiquitous. It's everywhere. I mean, just how much is important.
Yeah.
Is there.
So is that avian malaria will be following some seasonal patterns, or?
It's possible because there is some kind of relapse and recrudescence that happens. I mean, the parasite goes under dormant cycles. Then there is summer when there is bite rate of mosquitoes or vector increases, it goes again back up. So when you have very highly fluctuating seasons in other parts of India, where you have very dry and very cold or very wet seasons, then it may differ at what time you're sampling. It may give you false negatives. But in this site, it's very tropical, it's very aseasonal. And most of my sites were, my sampling time was around summers when the birds breed. They are immunologically compromised. Is the time when parasite will try to get into bloodstream. So I try to capture as much infection as possible.
And it's a very important question. Like Hawaii Islands, is there any like a study focusing on prevalence of avian malaria in Indian islands? Like for example, Lakshadweep has a colonization of mosquitoes. So is there any study going on, or?
No, we need samples. Nobody is letting us to go to Andaman Nicobar or Lakshadweep. Yeah. There would be, definitely. These, because there are mosquitoes in Andaman Nicobar for sure, a lot of them. There is human malaria. Yeah. You can take it outside. Thanks.